Here we are. My name is Philipp Harnich, and you can already see Barbara Imperiali here. Just give me a brief moment to introduce myself, introduce this masterclass, and then I'm going to introduce Barbara Imperiali, who's then going to introduce our speaker, Greg Dodge, today. My name is Philipp Harnich. I am here at QBiotech, head of laboratory. So this means I work every day directly at the bench, working with all our products, doing service for big pharma, for universities, and having really hands-on experience with that. I met Barbara Imperiali in Barcelona last year. We had a very nice chat, and this is how this collaboration and this wonderful masterclass came together. Here at Cube, we are experts for membrane protein research. We are a provider of protein services, but we do also produce consumables, meaning MAC beads and agaroses. This is very neat because we have hands-on experience in production and also in doing service, which means we can already go back to the production if we need some MAC beads, and then we can do very nice service. Here at Cube, we have the largest synthetic copolymer portfolio, which is available anywhere in the world. We have several different backbones working together with academia to get always the newest polymers on the market. And our scientific masterclass series, like this is today, offers researchers from around the world to present their latest research. And we are very proud to today have Greg Dodge here, and of course, Barbara Imperiali. She did her PhD at MIT in synthetic organic chemistry until 1983. Then she did a few postdocs and in the end got an assistant professorship at Carnegie Mellon University. After 3 years, she went to Caltech, where she had her first full professorship. There she stayed for 10 full years and then moved back again to MIT, where since 1999, she is now a full professor working with protein glycosylation, protein folding, thermostability, and also protein and peptide phosphorylation. And this is also where we met in Barcelona at the Gordon Research Conference, working with membrane proteins. And she and I were both very enthusiastic about the copolymers. And this is why I'm very, very happy to have her here today. So I will now give the word to you, Barbara. Feel free to start your microphone and introduce your wonderful poster.
Good morning, or perhaps good afternoon. I'm absolutely delighted to introduce Greg Dodge today. Just to say a few words, after completing his PhD at the University of Michigan in the labs of Professor Janet Smith, Greg decided to jump off the cliff from soluble proteins to membrane proteins, and he joined my lab in the biology department at MIT in 2018 with the goal of working with bacterial membrane proteins in glycoconjugate biosynthesis pathways. In the time that Greg has been at MIT, he has innovated and revolutionized all aspects of our membrane protein research. Some of this was in no small part due to the emerging SMALP technologies promoted by Cube, but the rest is in very large part due to Greg's limitless creativity and determination to develop efficient, robust procedures for tackling the structural and functional membrane proteome. He provides a huge service to all those around him, and I seldom see another person working at his bench with him as he shows them how to use SMALPs and other aspects of the membrane protein work. I think I should introduce Greg's talk as from cells to validated protein to grids, but I think in recognition of his EM pipeline, but most generally, Greg will tell you today just a tiny part of his accomplishments in his talk, which is actually entitled SMALPs as a Platform to Study Bacterial Membrane Proteins in a Native Membrane-like Environment. Native-like membrane environment. Thank you. Greg, over to you.
Thank you so much, Barbara. Just for our audience, of course, you feel free to ask questions. We have a very little chat window down at the bottom, and you are able to type your questions in there. And after the talk, we're going to have a question and answer round where my colleague is getting the questions one after another. We will put them on the screen so you can see them, and then Greg is going to happily answer all your questions. If we have so many questions, which I hope that we can't answer them all in time, of course, we're going to make sure that every of your questions is going to be answered afterwards. Okay, thank you very much. Also, Barbara, thank you, Greg, for being here. You can now start your camera. I'm going to put your talk on the screen, and then feel free to start. Thank you very much. We cannot hear you.
So let's dive right in. First of all, thank you both Philip for hosting, the folks at Cube for hosting, and Barbara for that awesome introduction. So yeah, today I'm going to share some of the research that I've been doing for the last few years in my postdoc here at MIT. And just as a brief overview for my talk, here's an outline of what I'm going to share with you today. So I'm going to start with a brief introduction about why we're interested in membrane proteins in the Imperiali lab. I will then transition to talking about some of the work that we've done for a particular target and share some of the methodology that we use to characterize that protein in a native-like environment. And then finally, I'm going to jump to what I think is really exciting I've been working on most recently, is trying to take the screening protocol that we had initially developed for this work and miniaturize it, and then come up with some more high throughput methods to actually characterize this miniaturized polymer screen. So let's dive in. So in the Imperiali lab, our interest in membrane proteins actually stems from bacterial glycoconjugates. And so why is that? So shown here on the left is an electron micrograph of an intact group 1 E. coli. And the reason that I really like this image is that it sort of highlights that really the first molecule that you encounter if you interact with a microbe is not the lipid bilayer, but it's actually a dense mesh network of heterogeneous glycopolymers. So that's what these sort of squiggles here are on the outside of the cell. And if we take a zoomed in view in this cartoon depiction here, you can see that there's, as I mentioned, this is a dense heterogeneous meshwork. There's a ton of different types of glycopolymers found on the outside of these bacteria. So over here on gram-negative, you can have things like lipopolysaccharides, you can have glycoproteins, lipooligosaccharides. Certain membrane-embedded proteins require glycosylation for function, like flagella. And then if you look at gram-positive membrane, you see this much thicker peptidoglycan layer, again, glycopolymer, but then you also have these other unique moieties like wall-teichoic acid and lipoteichoic acid. Now, the reason that these things are important, there are multiple reasons that these molecules are important. First, they're key for immune recognition, so determining whether a bacteria is pathogen or commensal happens at this level. And then also these are virulence factors, and they're important for overall stability of the organism that has these on the outside of them. So if you knock out the pathways that make these glycopolymers, that can be extremely deleterious for the bugs. And in fact, that represents one methodology for designing antibiotics. So if you can, for instance, shut down the production of peptidoglycan, you have a pretty decent chance of whatever compound that does that being a good antibiotic. But if we really want to understand, so if we want to take a look at this and say, okay, can we shut down the production of a specific glycoconjugate? We really have to understand how those glycoconjugates actually get produced. And this is where the situation gets pretty complex. So if we zoom in and take a look at, for example, the N-linked glycoprotein that Campylobacter jejuni, or the N-linked heptasaccharide that Campylobacter jejuni appends onto glycoproteins, it's pretty complex chemistry. So you have a series of individual monosaccharides that are linked in a specific way, and both the order of these monosaccharides and the stereochemistry of their linkages are important for function. And unlike any other biopolymer, these are not templated. So you can't just look at the genome and say, aha, that's what the sequence of this glycoconjugate is going to be. You have to actually study each individual enzyme that's responsible for adding these sugars one by one in order to A, figure out what particular sugars go into a glycoconjugate, unless you do mass spec sort of late stage. But then B, if you want to actually inhibit a particular enzyme or a particular step here, you have to be able to study that individual enzyme. And this is really where our interest in membrane proteins starts. If we take a look at how these pathways actually function, here is an example of a prototypic glycoconjugate biosynthesis pathway. So we're looking at the LPS O antigen biosynthesis in Salmonella enterica. The first thing that I want to point out is that each and every protein involved in this pathway is membrane embedded. So that raises the bar in terms of the difficulty for studying these pathways. The second thing that I want to point out is that across all of these steps, these chemical steps, there's one conserved player, and that's this undecaprenol phosphate. This is a very low abundance lipid, but it is critical in essentially all glycoconjugate pathways in bacteria. So the way that this works is you start with this undecaprenol phosphate. An initial enzyme, a class of enzymes, phosphoglycosyl transferases, will take a phosphosugar off a nucleotide sugar donor, append it onto this lipid, and then a series of glycosyl transfer reactions will happen where individual sugars get appended to this growing glycoconjugate. Once a particular length is reached, a flipase will translocate the nascent glycoconjugate across the membrane into the periplasmic space, where then a polymerase and a regulatory protein will polymerize these units together into a long set of chains, and then it will go on to be, in this case, appended to lipid A. Now, what we're interested most specifically in the Imperiali lab is these class of proteins, these phosphoglycosyl transferases. And we think they're fascinating enzymes, but from a practical perspective, we're really interested in them because they're the class of enzymes that catalyze these reactions in prokaryotes is unique. They don't exist in eukaryotes. So if we think about this pathway from the lens of inhibitor development, shutting down the beginning of these pathways represents an ideal way for stopping production of these glycoconjugates. And they're also very specific for their substrate. So if we can exploit that specificity, we may be able to shut down particular glycoconjugate pathways in particular organisms to have a very fine-grained way of attacking certain bugs. So what do we know about phosphoglycosyl transferases? Well, when I joined the lab, this is sort of where the field stood. So we solved the first crystal structure of this class of proteins in collaboration with Karen Allen's lab at BU. And this was detergent solubilized material, but we got this really nice crystal structure that was awesome for a few reasons. First, it was a unique fold. So that's always nice when you can put a brand new fold into the PDB. But then if we thought about the biology of what was actually happening here, it was a really interesting system. So we knew this protein was membrane embedded, but what we actually see here is that the way that it integrates into the membrane is by way of this reentrant membrane helix, which enters and exits the membrane on the same side. So you have a protein that is fully embedded into the membrane, but it doesn't span it. And that was really cool to see. And we learned a lot about this class of proteins from this initial structure, but if we take a look broadly at the superfamily of enzymes, what became obvious was actually there's a lot more that we still don't know. And what I'm showing over here is a different class of these proteins, which actually represents the majority of sequences that are available in say, UniProt. And what's different about this class of PGTs is that they actually have additional accessory domains. So there's a putative set of 4 transmembrane helices, followed by a domain of unknown function, and then a catalytic domain that is homologous to the crystal structure that we solved. So when I started, we really wanted to understand, okay, can we get more structural information of these proteins? And then more specifically, can we understand what's going on with this other class of PGTs? So this was the target set when I started in the lab. So we can see here we've got a variety of different types of PGTs. We've got proteins that use a different set of substrates, and then proteins that belong to different pathways. And again, many of the final glycoconjugates here are virulence factors. So it is, if we can figure out a way to shut these proteins down, that could be very, very useful for public health. So after about a 1.5 years of work, this is where things stood. Most of our success initially in the lab to get the crystal structure that I showed you was done using detergent solubilized material. And so following those protocols, I was only ever able to solubilize and purify the small PGTs by detergent. These large PGTs, which again represent the majority of the sequences that are out there, were totally recalcitrant to detergent solubilization, which was really frustrating. So, as we're talking, as the focus of this talk is alternative solubilization strategies, I just want to show this brief cartoon summarizing the ways that you can extract membrane proteins out of bilayer. So obviously we've got these sort of non-bilayer systems that are maybe the more classic way of handling membrane proteins, but there are downsides associated with things like detergent solubilization or using amphipoles. Namely, if there are important lipid interactions with your protein, you tend to lose them by extracting the protein away from all of the lipids. We're lucky that we now live in a time where there's ways to get around these issues. So there's tools like amphipoles, tools like the MSP nanodiscs popularized by Sliger. And then finally, we've got the focus of this talk, these styrene maleic acid copolymers, which allow you to make native lipo nanoparticles, extracting your protein directly from the membrane. So we were really intrigued by using these SMA copolymers to see if we could rescue some of our protein targets that were not soluble via detergent. But there were some hurdles getting there. So when we initially worked with SMA, one of the downsides that we ran into was that all of our proteins are expressed as sumo fusions, and we use sumo tag to help boost yield and stability during protein production and purification. And what we found actually was that, okay, we could solubilize our proteins in SMA, but if we tried to cleave off that sumo tag, which we wanted to get rid of for structural biology applications, this material was actually resistant to the sumo protease. And that was really frustrating, especially considering we had nice looking material going into it. And then, so we took a step back and we're like, okay, do we really need the sumo tag if we're in SMALPs anyways? And so we tried to just make his tag constructs of our proteins. And this is just a representative example. But if we just worked with his tag material, we would get high yields, but it was often very impure material. And it was super difficult to try to clean this up. And we really couldn't use this for structural studies. There's just too much other material there. So we went back to the drawing board and asked, can we come up with sort of a streamlined system that's focused on making our proteins and purifying them in lipo nanoparticles? And we came up with a scheme that, as I'll show you, works really well. But just to walk you through what happens here, we still use a sumo tag. We have a small linker, and then we switch to streptactin purification away from the his purification, which was giving us issues. We put in another linker with a TEV protease site in case we want to get rid of all of this, and then our protein of interest. And sort of the secret sauce here, in terms of getting around that sumo protease problem, is to express our proteins along with a second plasmid under an orthogonal inducing agent. In this case, it's under an arabinose PBAD operon, which allows us to make our protein first using IPTG or autoinduction is what we usually use, and then add in arabinose, which turns on production of sumo protease. We can cleave that sumo tag off before we ever add any polymer here. And then we can go ahead, this exposes our purification handle, and we can go ahead, extract our proteins from isolated membranes and get nice looking SMALPs, at least in theory. That's what we hoped would happen here. And when we put this in practice, we were actually thrilled to see this particular result. So shown here is a set of specifically large PGTs. So these are proteins which we could not extract using detergent, and a small panel of polymers. And as I've shown in red boxes here, we were able to get highly pure material. This is a one-step purification. So that was great to see. And in particular, we were really happy with this WbaP band, where obviously there's a ton of material there. It looks great. So if we go back and update that target table that I showed you before, if we switch to using SMA, we can actually solubilize and purify all of our targets. So at least for our proteins, it's obvious that these lipo nanoparticles are a much better system for extracting from the cell membrane. But everything that I just showed you was sort of small scale, more analytical. And we wanted to know, like, how far can we push this? Can we really scale up and do some hardcore biophysical studies of some of these proteins? And I want to focus today on WbaP, that protein from Salmonella enterica. So shown here is an SDS page gel from a scaled up purification. This is from a ½ liter of isolated, or cell membranes isolated from a ½ liter culture. And yeah, the purity and the yield, again, still looks great even when we scale it. So that was awesome to see. And in terms of the net yield, we can get higher than 10 milligrams per liter of this particular protein, which for those of you who work on membrane proteins, that's a lot. We were really, really happy with that. And then maybe one of the first real surprising results with this particular target was when we took this material and injected it onto a sizing column. First of all, we were very happy to see sort of the sharpness of these peaks. So this is very monodispersed material. But if we tried to calculate the molecular weight of the particles using just the calibration curve, we got 464 kilodaltons, which, given that this is about a 62 kilodalton protein, that seemed a little big. So we wanted to see how else we could look at these highly purified SMALPs. And so I'm just going to share some of the techniques that we've used successfully for this protein. So the first technique that we used was SECMALs. This has been done in the past for several different targets in SMALP. And really this gave us good confirmation that, yeah, the polydispersity of this protein is really low, and it gave us a much more reasonable mass range across this peak. So rather than the 400 plus kilodaltons that we were getting from our plain old sizing column, here we see a value of like 260-ish kilodaltons, which is a little more reasonable. However, there are some issues with using SECMALs when you have a multi-component system, and the mass value may not accurately be reflected because you don't necessarily know the differential refractive index value for each individual component. And especially given that there's lipid, protein, and polymer here, we wanted to see if there were sort of orthogonal ways that we could look at this. One of the ways that we accomplished that was using mass photometry, which simply uses light to measure the samples. And we were happy to see that, again, we got this mass range somewhere around the 240 kilodalton mark, which agrees nicely with SECMALs. And so right around this time, I met some folks from Thermo Fisher at a conference, and they were really excited to see some of the initial SMALP data that I showed. And one of the things that they were really interested in doing was trying to get native mass spec methods working with proteins in SMALP. And so this result here is really, really exciting because this is actually the first example of a protein embedded in a styrene maleic acid liponanoparticle flying on in a native mass spec experiment. So literally we just take our sample, exchange it into a volatile buffer, and boom, this is the mass that we get. So even though this is a pretty simple looking result, this is a really, really powerful technique that I hope folks who are working on these copolymer-based systems realize you can do this. And the protocol for this is published in our eLife paper that just came out recently. But all of what I've shown you up till now really only tells us about the full lipo nanoparticle. And it's a pretty large mass, again, considering our protein is fairly small, so 60-ish kilodaltons. And the first real experiment that we were able to do to get good data that informed us on the oligomeric state of our protein was lysine reactive crosslinking. And this is a little bit of a shot in the dark because it relies on lysines being close enough together at the oligomeric interface that you actually form a dimer. But we got lucky. So here's our an anti-strep tag gel. And as we go across our lanes here, we're increasing concentration of a lysine-specific crosslinker, and we can see a shift from a monomer to a dimer. The particular crosslinker that we chose is redox sensitive. So if we add a reducing agent, we can kick all of these back down to the dimer, or to the monomer rather. But then we wanted to take this one step further because the lysine reactive crosslinking just showed that there were some lysines present. We wondered if we could actually map this and get a readout for what the oligomeric interface actually was. So for that, again, we worked with our friends at Thermo, utilizing this T-butyl-FOX compound as well as this DSS, again, compounds that hit lysines to do cross-linking mass spec. We were able to see very distinct cross-linking patterns depending on the compound that we used. And then if we map those crosslinks onto an AlphaFold dimer model, we're actually able to validate that that model was a very reasonable prediction given what we saw in the cross-linking mass spec data, to the point where if we picked particular lysines that were present at the putative oligomeric interface, the only time that their cross-linking distance was satisfied, given the distance allowed by the cross-linking compounds, was when we had it modeled onto our dimer. So that was really awesome to see. There's a lot more data in the paper that I wish I could go into, but we were also able to use CryoEM to directly visualize this. So again, CryoEM has been used on proteins in SMALP, but I just want to highlight that we're talking about a protein that's 120 kilodaltons here. So pretty small, even by cryo-EM standards. And then to add on to the fact that there's a bunch of sort of noisy lipid and polymer around the outside of our protein, I think it's really cool that you can actually use this technique and this platform to do this. So shown here is our cryo-EM reconstruction at a threshold that allows us to see the lipo nanoparticle. So we can see that we've got sort of those 4 transmembrane helix domains bunched in the center, and then the domain of unknown function on top, and our catalytic domain as these sort of wings that are out on the side. And if we adjust our contouring, we can see, even though we're at relatively modest resolution, we can still see that our model fits really, really well. And again, the cross-linking data that we have agrees with this. So as I mentioned, there's a lot more data in this paper, but that's not really the focus of this talk. Here, I just wanted to show you some of the techniques that we've successfully employed for a particular target. So that's where I'm going to finish part 1. And I just want to summarize, these in-solution methods are really useful, and you can absolutely apply them to proteins isolated in these SMALPs. The big thing that I want to highlight, again, a huge shout out to the folks at Thermo, Rosa Wiener's team, for getting this technology working, but you absolutely can do direct mass spec on proteins isolated in SMALP. It works really well. And then, yeah, techniques like cross-linking and cross-linking mass spec can allow you to validate oligomeric state, even in the absence of primary structural data. So you can use an AlphaFold model and cross-linking mass spec, and in many cases be good to go. So now I want to jump to part 2 of the talk, which is sort of all started around this question of, okay, this works really well for one target, but what if we're interested in a lot of different proteins, membrane proteins, and even more broadly speaking, the membrane proteome in bacteria is pretty diverse. So shown here are just examples of membrane proteins involved in glycoconjugate biosynthesis in bacteria. There's a lot of different strategies for anchoring these proteins to the membrane. And we wanted to answer some fundamental questions. A, can you actually isolate and purify all of these different proteins in SMALP? But then B, how are we really going to do this from a practical perspective? So this challenge gets even more challenging if you take a look at how the polymer field has advanced recently. Where first I showed this data for these 4 polymers that we were using for screening. Now you can easily call up your buddies at Cube and end up with 16 polymers, and then here 10 plus targets of proteins. And it's very overwhelming, especially when you consider that if we were to operate at the scale that we did for the large PGTs, each one of these screening runs would use a half-liter culture worth of isolated cell membrane just for the initial screen, let alone you would burn through a ton of polymer. So really what I wanted to do was come up with a way to miniaturize this so that A, I wasn't having to grow tons and tons and tons of cells, and then B, we could conserve some of the polymers. And so really where this really took off was when I came across the cutest little benchtop ultra centrifuge that I've ever seen. So this is actually the rotor for it here. It's a TLA 100 rotor. And this allows us to shrink from a 7.5 mil scale for our initial polymer screening down to 150 microliters. So huge savings in terms of the isolated cell membranes, and of course, in terms of the polymer that you're using. This protocol is going to be published as a technical note through Cube in the future, so keep your eyes out for that. But basically, we can take 75 microliters of our isolated cell membranes, 15 of our polymer, some additional buffer, let that rotate for an hour at room temperature for solubilizing, and then spin this down in this baby ultracentrifuge. And then the next question that I had was, okay, so like miniaturization of the solubilization could work really well, but how does miniaturization of the purification strategy hold up? Do we get the same level of purity that we saw running at those larger scales? And thankfully, the answer is yes. So shown here is an example of the protein that I've talked about earlier, S-enteric WbaP. And each one of these lanes represents an individual different polymer. And as you can see, first of all, the purity, when you are able to purify the protein, looks great, but there is definitely a different profile depending on the polymer that you're using. So that was really exciting. But just using SDS Page isn't really enough to judge the quality of the material. And if we were to try to use any of the techniques that I showed you before, the small volume that we have here of 30 microliters of elution is not really enough to do things like SECMALs, mass photometry, and all of the cross-linking mass spec type experiments all at the same time from the same batch of protein. So we wondered if maybe there was some instrumentation or something like that that could help us get around this problem. And it turns out that there actually is a really great tool for doing this. So this is a total shout out to the folks at Nanotemper for building this instrument. It is absolutely awesome. So this is a Prometheus. We turned to this instrument called a Prometheus Panta, which allows us to simultaneously measure thermal stability by nano differential scanning thermometry, dynamic light scattering. So this is a built-in DLS reader, and then back reflection to take a look at sort of aggregates in solution. The nice part about the DSF module in this instrument is that it actually uses native tryptophan and tyrosine fluorescence. It doesn't rely on like Cipro dyes. The other good things here, 10 microliters per sample. Okay, now we're in a scale that we can really just take material from our screening run, put it right on this instrument, and get a ton of data. And this has been used for many different applications for proteins, buffer screening, ligand binding analysis, even detergent screening for identifying optimal detergents for membrane proteins. And we were really excited to see, is this actually going to work for SMALPs? And just to show sort of some example data from Nanotemper for what we're looking at here, this is an application note that they have available online where we're looking at a particular GPCR solubilized in detergent. On top is the fluorescence data coming out from one of the channels that gets measured. And you can see there are sort of these lines with inflection points in them. If you plot the first derivative of those lines, you get these sort of plots that either have peaks or valleys. And anywhere that there's a peak or a valley, that's an inflection point in the primary data. And that represents thermal unfolding of the protein at that particular temperature. And so what this example shows is if you take this GPCR, add in some different ligands, you can see the stability of the protein change. So this is just an example application using this technique. So the first question is, how does this material look by DLS? So this is a ton of data, but essentially what we're doing here is going through that set of polymers that I showed you previously. And each one of these represents the DLS results from a set of duplicate measurements. If we overlay these values, we can see that there's actually some distinct differences in the size of the liponanoparticles that we're getting. So the key thing that I want to point out here is we see sort of two distinct sizes, with some polymers giving a mixture of the two sizes. So we see a population here around 10 nanometers in terms of radius, which is what's often reported for SMALPs. But then we also see these larger particles that are about 30 nanometers in size, which is way too large for what you would expect from a SMALP. And then some of these polymers give sort of a mixture of the two. So right away we can say, okay, there's some polymers that work well, some polymers that don't. So that's awesome. And if we tabulate this data by cumulant radius and polydispersity, we can very easily identify certain polymers that outperform others. The key thing that I want to point out here is this SMA30 polymer is what we used for the WbaP work that I showed previously. And the radius that we get here, 6.8 nanometers, is spot on with what we actually observe in our CryoEM experiments, which is about 120 angstroms across. So that matches perfectly. What's kind of cool is that even for some of these other polymers that seem to behave well, we can get different sizes in terms of the radius. So maybe we can use different polymers to get different size particles that might have more lipid in them or something like that. And it might be useful to screen a set of these at once, say if we want to do cryo-EM, to identify which ones work best. But the real experiment that I was super interested in asking was, can we actually do the DSF portion, or use the DSF portion of this instrument and see thermal denaturation of our protein directly in SMALP? And I was really surprised to get this result. So this is one of the targets that worked pretty well. And I'm just going to walk you through this data here. So these top plots are the two wavelengths that get monitored during DSF. And what jumped out immediately, if we look at, again, the first derivative plot here, is there is a distinct peak, negative peak, in this case at 53, which implies there's some sort of shift in the local environment of the protein at that temperature. But because this instrument also measures backscattering and DLS, this gives us ways to assess sort of the overall state of the lipo nanoparticle at the same time. And what was really, really cool to see is the accumulant radius does not change at this temperature. So whatever is happening here inside this lipo nanoparticle, it's happening to the protein. It's not happening to the actual particle itself. And it's not until you start to get to temperatures 70 plus that we see these particles starting to fuse into sort of larger, maybe aggregates, things like that. But it's not all perfect. So this was one example. If we look at some of the other polymers, the data is a little bit noisy. And I think probably what's happening there is that the yield for certain polymers is better than others. And it's really only once you have yield above a critical threshold that you get good signal. So even though we know SMA30 works well for this protein, maybe we see a thermal transition here at 53, but it's not super clear. However, just being able to see this one trace where it looked like we had genuine good DSF data was enough to say, okay, let's test this with some of the other proteins that we have. So now I want to jump to this protein here, Helicobacter pylori, PGLC. It's really small. Again, it sits in the membrane just like the first crystal structure I showed you at the beginning of this talk. When we put it through our polymer screen, just like we saw for WbaP, we see some polymers perform well in terms of making these really tight size ranges by DLS, and some perform poorly. However, if you were to just look at the SDS page gel, you would never be able to tell that apart. So by having this orthogonal way of looking at the cumulant radius of these particles, really is a powerful analytical technique. Again, we can highlight the ones that look good. And then really what was super cool about this target in particular was when we jumped to the actual DSF experiment. And now here we're seeing data that looks really, really good. So it doesn't matter which particle we're looking at here or which type of polymer we're looking at here. We see genuine unfolding that is decoupled from an increase in size or aggregation, implying that the protein is unfolding prior to the lipo nanoparticles falling apart. And in some cases, we even see multiple peaks, like here for SMA30. So maybe there's some sort of the unfolding trajectory may have some sort of intermediate that happens first here. But what was really cool about this, even beyond the fact that the data looks good, was if we compare the results for SMA polymers versus some of the other types of polymers, these aminoacrylate polymers, while they look great by DLS and they look great by SDS page, these lipo nanoparticles, or at least the protein in them, is 10 degrees less stable than SMALPs. So again, by using this technique, you could just straight identify, okay, this one polymer is best in terms of size, in terms of yield, and in terms of thermostability of our protein. So that was, I think this data is just really awesome. And you can do this all in an afternoon. It's great. And then finally, the last target that I want to talk about is this N-gonorrhea PGLB. The reason that I think this is interesting is that this protein has a large soluble domain that's actually attached to the PGT domain. And on top of that, we know it forms an obligate trimer. And it also has another sort of subdomain shown here. So we've got an acetyltransferase domain, a nucleotide binding domain. These are soluble. And then a PGT domain that's membrane embedded. The overall thing forms a heterotrimer, or a homotrimer, sorry. So again, we can put this through our polymer screen. Things I want to highlight here are the polydispersity is actually really low for some of these polymers, which is really cool to see. The gel is maybe not, or the SDS page gel is maybe not as nice looking as the other gels that I've shown you. It looks like maybe we've got a little bit of proteolysis or something going on here. But if we take a look at the DSF data, again, we see some polymers, doesn't look great. Some polymers, we see maybe a transition. But what was really cool was this acrylic acid polymer, ASTI 6-45, which shows very clearly multiple peaks on our DSF plot. And then actually the first time that we were able to see an increase in turbidity, meaning the protein was crashing out. And it was cool that this correlated with the only protein that had a large soluble domain. So multiple thermal transitions, the protein is crashing out. And this all sort of lines up nicely with what we know about what should be in these SMALPs. And then sort of the last bit of teaser data that I want to show for this particular target is that if I take sample directly out of this elution buffer, so there's still biotin around in this case, and plunge a grid, a cryo-EM grid of that material, we can actually get pretty decent looking particles. I'm not sure exactly how that looks for you guys with the compression of the PDFs and stuff, but we were really surprised to see that we might be able to go just from these small scale screening directly to grids, sort of maybe even in an afternoon. So this could be a really, really quick and efficient workflow. Okay, so that's it. So to summarize, we can run these. I won't say high throughput because it's not like we're doing thousands of samples in a day, but a moderate throughput nano DSF screen and really quickly identify, okay, this polymer is optimal for this protein. It gives a size of X. It has a thermostability of Y. And if you look at all of these different targets, which again have different topology, they're different classes of proteins, there is particular preference for certain polymers over others, and the sizes are not necessarily the same. But the one thing that we do see consistently is that these lipo nanoparticles don't really start to blow up, for lack of a better term, or form gross aggregates until a really high temperature. And I think that speaks to sort of one of the really nice things about these polymers is that, or about these lipo nanoparticles, is that they're very stable. And this has been reported, you can set them in the cold room for a week and your protein is still active, things like that. But to actually be able to assign, a melting temperature to this, I think is pretty cool. And will be useful to sort of study the different polymer systems that are out there and continue to come out. Okay. So that's it for me. I want to thank a lot of people. So obviously Barbara, it's been awesome. I've been in her lab for a little over 5 years now. It's been a great time. In particular, I want to thank her for just allowing me to pursue crazy ideas. None of this stuff would have been possible without her sort of trusting me and believing in me. And then got to thank all of the lab members for their support. In particular, Alyssa Anderson, who was a PhD student, is now working in industry, was instrumental in a lot of the WbaP work that I shared in the first part of my talk. Got to thank our collaborators in the Allen lab at BU. So they have some data that didn't make it into my talk, but will hopefully end up in their own papers soon enough. Got to thank the folks at Thermo for help with all of the method development and mass spec data processing. And then finally, Cube for putting this on, for inviting me, and for also sanity checking the initial DSF data. So I'm not the only one who has seen this stuff. Cube also has used this technique and it works for some of their targets. So with that, I think we'll jump to Q&A.
Okay, I think this fits. Great, thank you very much, Greg. This was fantastic. I loved it. And yes, I can sanity check you as you could sanity check me, because I saw exactly the same. Like, these polymers are super temperature stable. And I couldn't believe it myself, so I'm very happy that you see the same. While my colleague is fixing the questions, let me start with the first question. We are working much with like drug targets, GPCRs and stuff like this. And what we think is amazing at these polymers is that they are indeed that temperature stable. And I had stuff on my benchtop for like 3-4 weeks. The stuff was still same size. same look in sex, same profile. Like what do you think? Like, will these polymers get the drug targeting, GPCR, whatever? Will it elevate this? Do you think this is a real deal, or is it too good to be true?
Oh, that's a big question. I don't think it's necessarily too good to be true, right? I think the stability of proteins in these particles is probably not a question at this point. It's been reported over and over again. Now, in terms of will these really accelerate drug discovery, I think that depends on the particular target that you're working with and the particular question that you're trying to address. So the one thing that I am still curious about myself is, what's happening in terms of dynamics in these liponanoparticles. Are the proteins able to sort of flex like they normally would, or are they rigid? And in particular for GPCRs, I think that's an important question, because obviously the small flexations in those proteins is really important for how they function. So you want to make sure you're sampling the correct structural subpopulations for like drug development. But one way that you might be able to test that is by using conformationally specific nanobodies and seeing if they still bind the proteins in SMALP. That might be the way to go to at least give yourself the confidence that you're looking at what you think you are.
Yeah, great. That's wonderful. I think it's exactly the same. So yeah, it's really important that the protein stays flexible, that you have the lipids around, that it's correctly folded, and that it withstands like all the environment factors there. Okay, Sebastian, are you ready to start a question round from?
Yeah, let's do it. Yes,
that's the first question.
Honestly, this question has haunted me, right? so do the styrenes not interfere with DSF? It seems like they don't. That's the only answer that I can give, is that like we've looked at this with a lot of different proteins. The folks at Cube and polymers, the folks at Cube have done the same. And across the board, whatever thermal transitions we're seeing sort of before the SMALPs blow up seem to be dependent on the protein that's inside the liponanoparticle, and then that large increase in overall particle radius seems to be the particles either maybe fusing together or something. I'm not exactly sure physically what's happening there, but yeah, it really does not seem like the styrenes interfere much, which does make sense, because a tyrosine, the particular wavelength that you're measuring tyrosine fluorescence at is not necessarily a wavelength that styrene interferes with strongly, right? It's more towards the 350 nanometer as opposed to 260.
Okay, can we use these polymers with insect and mammalian cell lines too for the membrane protein isolation and purification?
So I think I'm going to defer to Philip for this one. So all of the data that I've shown today are out of bacterial membranes. And every time I talk to people, they're like, well, I work on mammalian cells. Like, can I do this? And I can only say, like, I think you're supposed to be able to. But I think Philip may be a better person to answer that.
If I may, I don't want to steal the show from you. But yes, absolutely, you can. Like, I personally done this with more than 30 targets, and it works wonderfully. It's important that you do the screening. I mean, I don't want to advertise too much. We have the screening kit. There's all the polymers in. You can do this very fast with your wonderful protocol. As you said, we're gonna put that on our website with an application note together, so that you guys have all the info there. But yes, absolutely you can. And there's no limitation. Channels, GPCRs, whatever you wish, it works in dramatically many cases. Like, there's not really anything which does not work out with that. If you can express it, you can purify it. Okay, how do you confirm that the native protein conformation is good before doing denaturation analysis?
So I think this gets back to the first question, Philip, that you asked, of like, what really is the state of the protein in solution? And to answer that question, we don't necessarily do anything directly, but what I can say is that we do have an activity assay in particular for the PGTs, and our proteins are active in SMALPs. So we know that at least they're still capable of catalyzing the reaction that they should. I guess if we really wanted to, we could like look at them by CryoEM before we do all of this, but we try to think of the denaturation analysis as sort of a front end QC. But we do know, again, for just the PGTs right now, that the proteins are still active, and that's probably the closest that we can get to native protein conformation being good.
Yeah, and we see the same for mammalian and insect purified proteins. Like we also do activity assays, and as you said, it's a screening approach to see what is the best fit, and then you go from there. Okay, are you solubilizing directly from the bacterial cell pellet, or do you first need to lyse the cells and prepare an infection? It's a good question.
Great question. Awesome question. So for all of the work that I've shown, these are from isolated cell membranes that we get using differential centrifugation. I know there are reports out there that you can go directly from like a bacterial cell pellet, but we like to sort of simplify the system. So by getting rid of all the soluble stuff and focusing specifically on isolated cell membranes, it makes our life a little easier. It also enriches targets. So if you have problems with overall expression level, by isolating the cell membrane first, you can boost the effective concentration and get around yield problems.
Yes,
so I do highly recommend that.
Absolutely. If you're working with bacterial proteins, there's often the issue of the proteoglycan, which the polymer cannot penetrate so well. So it's better to open the cell first. But if you're working with mammalian or insect cell lines, you can absolutely go directly from the cell. We do that all the time. Okay, let's do two more because we're already a bit hot on time. When you measure the mass with SMALPs, do you get the information about protein mass only or about the complex of protein and SMALPs?
Great question. As of right now, we get mass information about the entire particle. So it's polymer, lipid, and protein. If you want to get information about the mass of protein only, you may be able to do that by, in particular, if you're using SMALP, by adding in a large concentration excess magnesium to destabilize the SMA polymer in the presence of detergent to strip away everything else, and then looking at it by mass spec. But obviously there, you've gone away from sort of the advantages of this native system. So I think you can do it, but at least most of the data that I showed right now are total intact particle. Yeah.
Okay, one last question. Is there a way to perform TPP, SETSAR, or PISA on mass spec and drug discovery screen for drug compound effect on membrane protein thermostability? Well, that's a long question.
Wow. I'm actually not familiar with these techniques, so I don't feel like I can answer them. Yeah, Philip, do you have any insight?
Oh, honestly, I have absolutely no clue. No, but we're gonna do our best to answer the question afterwards via email. Then let's do one last to end with a good one. Did the exact excess of biotin in your elution buffer affect the biophysical analysis?
Good question. It doesn't seem like it does. So to be absolutely clear, all of the stuff that I showed in the first part of my talk, the scaled up experiments, we put that through a sizing column that removes the biotin. Now that in terms of quote unquote excess biotin, it's only 50 millimolar. It's not a ton. So it's roughly the same concentration as buffer components. And we don't see really an issue with that in any of the screening type analysis, whether it's SDS page, DLS, things like that. And in fact, I was surprised to see that when we plunge grids of the material that still has biotin in there, it looks pretty darn good. It's not like it affects the ice quality in CryoEM either. So, it doesn't seem to be hurting the material, but you could do something like a microsizing or one of those little tiny dialysis buttons to try to get rid of the biotin if you're really concerned about having it around.
Yeah, exactly. Wonderful. Thank you so much. It was great having you. I'm very happy we were able to arrange that. And of course, all the questions we will not be able to answer right now. We're going to make sure that you get a response via email. The new protocol, the application note is going to be on our website as soon as possible. We are also going to inform all of you, all participants, and also everybody on the website, so that you will not miss this. And yes, thank you very much. And I nearly forgot. Thank you, Sebastian. The next masterclass, if I can end this with a little bit of advertising, is going to be held by my colleague Michael and myself. It's going to be already in 2 weeks, and we're going to talk a bit more broad about the whole copolymer solubilization, what kind of classes are there, how do they differ, how do the backbones differ, and what is the right polymer for you and your specific target. This is what we're going to address there. So feel free to register, and yeah, maybe see you around in 2 weeks. Thank you very much, Greg, and you have a great day.
Thank you guys for inviting me, and thanks everyone for listening.